Hey everyone, and welcome to our videos for our second lab of Bio 201. My name is Julia Nitchman, and this set of videos is going to continue on a lot of the skills and terminology we learned last week, going over epithelial histology, but now we're going to look at those three other major tissue types: connective tissue, muscle tissue, and nerve tissue. This week's set of videos are going to include a brief kind of recap overview of some important things about the microscope, important tips for microscope use, and then we're going to dive right into those three last major tissue types that we didn't get to last week. So our big goals for this week are going to be able to identify the components of and use a microscope, skills that we should have already been building last week in lab, but that we'll continue to refine and practice this week. The ability to identify various cell types and understand why they are located where they are based on their function. Last week, we only dealt with epithelial cells. This week, we're going to deal with different types of cells, things like chondrocytes, osteocytes, erythrocytes, things like fibroblasts. So we're actually going to be talking about several different cell types instead of just the one overarching epithelial cell type that we talked about last week. We're also going to want to be able to identify the various common aspects of these cells and the structures they create. This is very important, especially in like connective tissue, where our cells are actually going to be helping to create an extracellular matrix around them. We're going to want to be able to identify the unique features of each tissue type, and then we're going to want to be able to identify the histological images from the presentation on the microscopes as well as APR. So a lot of the same goals, actually kind of all the same goals as last week, just instead of dealing with just one of the four tissue types, now we're going to round out and learn about all four of our basic tissue types. So before we jump into the new information, let's just briefly revisit some important info about the microscopes. Again, there is a link here on the slides for you to go watch a video if you would like a refresher on how to use the microscope and sort of the anatomy of the microscope. I just want to reiterate, when you get to lab, it is always best practice to start on the lowest objective lens, that 4x objective lens, since it gives you the widest view. Again, if I want to go bird watching, I don't go to the park, sit down on a bench, and immediately start looking through my binoculars at the trees, because I'm probably never going to find a bird that way. I'm too zoomed in. So you want to use your regular eyes first, that widest objective lens, the lowest power one, and then you want to get the slide where you can see something already. So you can see some colors that you're looking for. It might be a tissue that you're able to see some structures already at that 4x objective power. And then you're going to want to start increasing the objective lenses to the 10x and the 40x from there. You never want to start at that most zoomed in level, because you're going to be searching that plate all over all class trying to find what you're looking for. So always start wide, start with your eyes at the park, find the bird, and then put the binoculars on and start looking more closely, more zoomed in. Remember that in this lab, we will never use the 100x objective power lens. For most of the microscopes, that's going to have a little piece of red tape around it. That is an oil immersion lens. We do not use oil immersion mounts in our lab, so we never have to use that lens to look at our slides. And then as a recap, many of our slides are going to be mounted using a hematoxylin and eosin stain combination, also known as H&E stain. This is the most widely used staining technique for human histology. Remember, it is the type of stain that is going to stain DNA purple and proteins pink, and lipids are not going to hold on to any of that stain. So for many of our tissues, it is really useful to see the protein and the DNA, so this is a great stain option to use. So a lot of the tissues you'll see in lab this week are going to use that H&E stain. And so if you're seeing stuff that's pink, you know that's proteins. If you're seeing stuff that's purple, you know that's DNA. Not all tissues are going to use this staining technique, though, because sometimes we want to see structures that are not going to stain well with this technique. For example, if we're looking for very specific structures like the intercalated discs in cardiac muscle, that's not going to stain so well with the H&E stain. So we're actually going to use an entirely different stain for that tissue so that it highlights those intercalated discs. So you'll notice that cardiac muscle tissue, which muscle you might expect to see pink for, that is actually going to look blue under the microscope with our slides. That's done because that stain is going to stain the muscle tissue blue, but it's also going to very darkly stain those intercalated discs. So, and you have to kind of remember that We can use all sorts of different stains for our histology mounts, and the stains we choose tend to depend very heavily on what we think is most important to see in that tissue type. So there will be times where, like, the images we have on the PowerPoint of the microscope slides may be different in coloration and staining from the actual microscope slides we have in lab. That being said, the vast majority of our mounts use that H&E stain. So for the vast majority of our tissues, if you're seeing pink on your slide, you know that's proteins. If you're seeing purple on your slide, you know that's DNA. If you're seeing clear areas through your slide, those are either things like the lumen, where there is nothing, or it might be areas of lipids, like in these adipocytes we see here, where we have this huge area where adipocytes are essentially just sacs of lipids. So you get the pink of the cell membrane, the purple of the nucleus that's shoved all the way to the edge of the cell, and then that large amount of lipids in the center of the cell. All right, so that was a very, very, very brief overview of things to expect this week. I'm going to go ahead and start a new video here just so that all of the connective tissues are contained in one section of videos, or one video, I should say. But I look forward to seeing you in the next video, where we actually start to jump into our new tissue types, starting off with connective tissue. Bye.